Jesse Rose, and our Evolution Ecology Lab project is over plant defenses and herbivory.
We'd like to thank Dr. Paul Kowinski for letting us use his backyard and tomato plants as our experimental subject and site. Jenny Schaefer for letting us use the leaf scanner and written material. Professor Nickel for helping us out with data analysis. Then the biology department for letting us use materials in the lab space.
So when you imagine predator-prey interactions, typically you'll probably think of something more along the lines of this. But the truth is, it can look like this, but it can also look just like this in the case of insect herbivory. So predation can be broken down into the following sequence. The search, in which the predator has to actually search for the prey. The recognition, in which the predator recognizes that something that it finds is something that can in fact be eaten. The capture, in which the predator actually gets a hold of its prey. And the handling, which involve the actual process of killing and eating the prey. So unlike animal prey, plant prey can't defend themselves during the search, recognize, or capture phases of the predation sequence because they can't run, they can't hide, so instead they have to defend themselves during the handling phase. So plant defenses can be broken down into two main categories, physical and chemical. Physical defenses include spines, thorns, things you imagine on cactuses, whereas chemical deterrents are more subtle. These are secondary compounds produced by plants that might be toxic, they might be bad tasting or bad smelling, they may be indigestible, and trichomes are small hairs on plant surfaces, and they can actually be both physical or chemical.
Another way to look at trichomes is by looking at them like they're archer stakes. Archer stakes are sharp wooden stakes that are impaled in the ground at an angle. They're used to deter enemies from reaching the castle walls, which house all the valuables. Trichomes do the same thing. They deter the herbivore from crawling up the stem and reaching the leaf.
Now, trichomes themselves can be broken down into two groups, glandular and non-glandular trichomes. Glandular trichomes have a little bulbous end at the tip, which contains some sort of chemical right down the left, and non-glandular trichomes are more just straight and pointed at the end. Now, the little graphic on the right shows just how many different types of trichomes there are, and many of them can serve different functions. The particular type of trichome present in this study are glandular trichomes.
So previous research on the effectiveness of trichomes against herbivory has been somewhat ambiguous. In the Carly et al. Paper in 2015, they found that plants with higher density of trichomes did repel two-spotted spiders, which is one herbivore, but they did not effectively deter aphids, a different kind of herbivore.
The Riddick and Simmons paper showed that predatory insects can be trapped or impaled in trichomes, particularly on tomato plants, but there was still some ambiguity about the real function of trichomes. So this led us to ask the question, will the presence of trichomes reduce the amount of herbivory on leaves, the amount of trichomes on the stems of plants, reduce the amount of herbivory on the leaves of those stems? And we hypothesized that yes, presence of trichomes will reduce herbivory.
Okay, so when it came to determining the variables of this experiment, we first selected tomato plants as our prey for this herbivory interaction based on not only the plant's seasonality, but also the plant's availability, and it just so happened that Dr. Kowinski had an abundance of this plant in his backyard. From here, we then selected our predator, the hornworm caterpillar, which is a well-known consumer of the tomato plant. So when it came time for experimental setup, we selected 10 tomato plants, 6 of which were used as a control, meaning their trichomes were on the stems, and 4 of which were used as experimentals, and their trichomes were removed from the stems. When it came to removing the trichomes from the stems, we studied various methods underneath a microscope and found that, in fact, the best way to remove the trichomes from the stems was to scratch and brush the trichomes themselves off of the stems. And then after this was done, we isolated the 3 terminal leaves on the ends of each stem, and in total this would be 10 plants. And this provided the caterpillar with a variety of leaves to choose from. And then once these 3 leaves were isolated, we then traced and outlined the border of each leaf. And from here, the enclosures were then made, and We used Tool, which is a mesh-like material that mimics the natural environment of a field experiment while maintaining the caterpillar to its respective plant. And in addition, we used a cone and placed it at the base of each stem in order to ensure that the caterpillar was not using the enclosure itself as a means of avoiding the trichomes. But this created a direct path that the caterpillars had to crawl. And then from here, the enclosures were then finished, and one caterpillar was placed on the base of each stem, and the enclosures were tied. We then gave the caterpillars 24 hours to feast, and we came back the next day to retrace and re-outline each of the 3 terminal leaves in the same way that we had done prior. From here, both the pre and post application tracings were cut out, and a leaf scanner was used to measure the surface area of both the pre and post application tracings. And when it came to the measure of herbivory, the difference in surface area between the pre and post application tracings was an indicator of the amount of herbivory.
Now, to analyze our data, we first had to test for normality, and through Shapiro-Wilks tests with p-values of 0.85 and 0.38, we found that both of our groups were normally distributed. And then we used a Levine's test to determine if both groups had similar variance, and we found that yes, both groups did have similar variance with a p-value of 0.24. And then to determine if the 2 means we found were different, we used an analysis of variance, and we found with a p-value of 0.832 that the 2 means were not different. In the graph shown, the dots are our means and the bars are our 95% confidence intervals.
So to restate, our hypothesis was that trichome removed plants would have the most area consumed by an herbivore, but we found out that removing the trichomes did not affect the amount of area consumed on a leaf by an herbivore.
Some biological explanations that we came up with was that because we weren't able to starve the hornworms, they probably weren't as hungry because they weren't starved, so they didn't eat as much. And then the location of the trichomes and the area 1 on the screen there is the trunk, and you can tell that it is more trichome dense. And the area 2 on the screen is the stem, and it has lesser and thinner trichomes, and we could have put the herbivores on the trunk instead of the stem to help get a better explanation. The oils, whatever we were removing, the trichomes, we most likely got skinned oils on the leaves and the stems, and that might have deterred the hornworms from wanting to consume any of them. And then the trichomes are a synergistic defense, which means that they are most likely, or they are only one type of defense that a plant has to protect itself.
So our conclusion was that, consistent with previous research, we can't make a clear claim about whether trichomes function effectively to deter herbivores. But we do have some ideas about some improvements and further study. So we could perform this in a lab. We did this in the field, and there were a lot of variables that were hard to control, including temperature. Getting towards the end of the year, it definitely got colder, and we think that also may have played a role in how much the caterpillars ate. We also could increase measurement consistency. We had some difficulties getting consistent measurements with our leaf scanner, so we could retry another method with this. And then lastly, we could replicate this whole thing with a different plant and a different herbivore. A plant, we could have used something that had more trichomes than what we ended up with. And then for we could try different herbivores, because like in the Carly et al. Paper, trichomes may be effective against some herbivores, but may not be effective against others. Some practical applications of doing further study would have to do with contemporary agriculture. More and more contemporary farmers are trying to rely less on chemical pesticides and insecticides, and may be worthwhile to start investigating how to boost plants' natural defenses against crop pests. This is our literature cited, and it's have questions.
So you say you did 3 different statistical tests, and so
Yes.
Uh,
The first 2 were just to allow us to be able to do the analysis of variance.
okay, so you did the first 2 just to get to the third step.
Yes.
Okay.
Can you like re-explain the thought process behind like the starving explanation?
Yes, well, the thing is, we actually tried this experiment. We tried a first trial in which we did starve our caterpillars. And the thing is that in that process, we killed a lot of them. A lot of them died from the starvation period because we think it was too long. But the ones that did stay alive, they ate considerably more than our caterpillars the second round. So ideally, we would have liked to have a starvation period, but it was kind of risky. But anyway, to starve the caterpillars from the food that they were eating in the lab setting would have maybe upped their herbivory outside in the field.
But like, would that have upped their herbivory on like trichomed areas and untrichomed areas?
We think so. The first time we did the trial, there was a considerable amount of the leaf area gone, and we were like, okay, like this is great. Hungry little caterpillars, you know, they just ate through the leaves. But the second time, there was, between both no trichomes and trichomes, there was almost no herbivory, and we thought this is possible that this could have been due to.
And the problem with that first one is when we went back to measure with the leaf scanner, we tried just doing just the leaf scanner on the leaves themselves without tracing them, and that was incredibly inconsistent, and like we could tell visually that there was area eaten out of the leaves, but the leaf scanner was not picking that up, so we had to do it again.
The surface area actually increased from
Okay.
the first measurement to the second.
Were they the same caterpillars that you used in
Yes,
that
they were.
preliminary?
Yeah, they were just older
How
and
long?
much larger.
It was a week.
They were the same exact ones?
Yes, same
Okay.
exact cat.
They just grew in size because they had.
How long does it take them to digest food or need to eat more? I don't know. Did you guys?
They pretty much eat constantly while they're in the caterpillar stage.
Okay, but is there a period when they stop eating? Could that have explained why maybe like?
They don't stop until they start to pupate. They just
Okay.
eat constantly and they keep growing until they pupate. And these are moths, so until they
Yeah.
turn into moths, they're eating.
Did you guys keep them or did you guys get rid of them?
We tried to allow them to pupate, but we came back from Thanksgiving break and they were all moldy and dead.
Were they in the bag the first time you did it as well? Like the same apparatus?
same exact, yeah.
The only thing that was different was you just didn't starve them.
And obviously
And
the
the
temperature.
tracing.
And
Yeah, the tracing.
Yeah.
Why didn't you guys do it in the lab? Like, why not?
I think because
We would have had to grow the plants. We would have had to go through that whole process. So Dr. Quincy kind of suggested, he's like, well, we have these abundance of tomato plants already in the field in my yard. So that's kind of the convenience route that we went.
Yeah, if you're having to grow your tomato plants, you'd still be working.
Come on, pressing
They'd be
the
this
button.
big.
Anything else? Yeah.
You mentioned synergistic defense. I'm just curious, what was that again? I entirely missed that.
It means that like trichomes are only one defense that a plant has against insects from eating it, and so plants have a lot more than that, but that's just like the one that we focused on.
Yeah, like plants can have multiple layers of defense, like chemical and physical, and trichomes may just be one or the other, and they may not be effective against caterpillars particularly, whereas another defense that a tomato plant may have could be more effective against caterpillars.
Anything else?
Yeah.
This is kind of a hard question. Why does this research matter? Because it seems like you are just doing something similar to what previous researchers had done, but with different plants and predators. So what makes it different? What makes it important?
Okay, so I know that one thing that's important is we briefly touched on it, but the agricultural industry, being able to control herbivores and pesticides, or without the use of pesticides, can change the way that agriculture is done. And if we can discover a way effectively use trichomes or remove trichomes, how basically discover what the effect this form of defense has on its herbivores may lead to future progress in a healthier or cleaner world of agriculture.
We also, a lot of the previous studies just tested different kinds of plants, some that had naturally more dense trichomes and that some just had naturally less. What we did is by basically taking, instead of doing a measure of density, we just did presence or absence. So it was much more dramatic difference.
So is there like a realistic way that you can like get plants to grow more trichomes? Or like
I read
or
in,
less?
while researching trichomes, I read that there's a certain gene or chemical that causes the growth of trichomes, and this can be altered in plants. I don't have like exact
Yeah.
data on it, but yeah, the growth of trichomes can be controlled, I believe.
When you fasted your caterpillars the first time, did you give them water?
We did not know.
That may have been why they died. Not food, but
That's true.
water.
How long was that fasting period?
24 hours.
That would be bizarre. In your introduction, you referred to spiders as herbivores, which I found intriguing. In that study you were talking about, were the spiders playing the role of herbivores, or did they have a different role?
I could be mistaken on that. I maybe categorize them as herbivores, but they actually might have been just studying the effect of the trichomes on populations of insects in general. From what I understood.
What was the other species they were looking at in that study?
Aphids.
So what might the spiders be doing in that case?
Preying upon the aphids.
So if trichomes affect the occurrence or mobility or anything like that of spiders, what might that mean for the aphid population?
That it would grow.
Go ahead, Anne, if you have something. I thought I had something else.
Intro slide before you have the background information, or the background studies.
Sorry.
I don't know if I missed it or not. Can you tell me about the second study? I don't feel like I remember hearing about that one.
No, that one was about like just whether or not the study itself was about whether trichomes were harmful or helpful to other insects that may, because insects can just live in trichomes, and they found that trichomes were more harmful because they can trap or impale small insects, such as like lacewings, particularly, were impaled on the trichomes. So that's one study about the effect they have on other insects, but not necessarily the effect they have on herbivory.
The trichomes of the tomato plants, I maybe missed it. Did you say they were chemical or just physical? Because you mentioned
They're glandular, which can form as, they can function as both, because they are a physical
Basic
structure,
But
but
they-
they
yeah,
do
they-
contain
they secrete
chemical as
things.
well. Yeah. And we're not sure what chemical is in tomato plant trichomes or what that functions as.
Are trichomes an induced response to herbivory or not?
No, I believe they're natural.
What do you mean natural?
They'll grow in the absence or presence of herbivory.
Who drew your cartoons?
I did.
Non-sciencey question, but I was curious.
What were some of the different ways you tried to remove trichomes, and why did you ultimately decide to use the method that you did?
We looked at them under a microscope, and we actually used several different methods. We tried Scotch tape, like literally like a waxing treatment on our stems. We actually tried masking tape and Scotch tape. We tried burning them, which singed them, but didn't always get kind of the close little nubs. What would the couple of the other
Shaving.
ones?
We tried
Shaving
shaving them
them.
with a razor. laser, and we found with both the Scotch tape and the shaving that damaged the stem itself. And we found that not only was rubbing it with our fingers and nails the best way to get the trichomes off, it also damaged the stems the least.
There was still some damage, right?
A little bit,
I
but
mean,
very minimal.
Especially.
Relatively,
Relative to the other method.
Yeah.
Yeah, so it was like literally
Yeah,
big
shaving
salad.
it like we almost cut the
Oh.
whole stem. So
Right.
just hard to control.
I'm done whenever you are.
Let's see, in the discussion section, you mentioned that you could use a different method for measuring the loop area. What might that be?
Well, you guys mentioned in the process of doing kind of like, basically putting a clear kind of piece of material like plastic or glass, and then kind of putting dots on it to measure the amount eaten basically from beginning to end by like quantifying the number of dots for each leaf. And that probably would have been a better route to go. The leaf scanner seemed pretty promising in the beginning. I can't even really entirely tell you why we had so many problems that we did, but we think that there could be potential other ways to quantify that rather than use the leaf scanner.
We could also probably have used a digital program. I don't know if it's out there, but allows us to take a picture of the leaf itself and measure the difference in surface area like electronically based on like pixel size.
Yeah.
I think you actually mentioned that, but we didn't know if we could feasibly get it
Find it,
consistently
yeah,
enough,
yeah.
like taking it from the
Right,
exact
to get the right
distance, because that has to be
distance.
very precise
Yeah.
for pixel counting.
That could be a little more challenging in the field.
Yeah.
You have to attach things to your camera to make sure that you're maintaining the same distance all the
Exactly.
time. It can be done, it's kind of a pain in the butt, but so is using a leaf scanner that gives you
Yeah.
nice results. It's one of those situations where my experience is a lot of times in the field, the lower tech option is oftentimes the most repeatable